Hello students, welcome to M.Sc. Microbiology course CCTP 7 Immunology. Today we are going to discuss about T-cell receptor from the chapter Cell Surface Molecules and Receptors. T cells, they are so called because they matured in the thymus. B cells, for their activation and proliferation, they require a signal which was provided mainly by T cells. So these T cells, when they recognize the antigenic peptide which was presented by antigen presenting cell, they start to secrete the cytokines. Now these cytokines, they bind to the receptors which are present on the B cell, and B cells start activating and proliferating. Once they are getting proliferated, they produce effector cells and the memory cells. The major function of the memory effector cell is to produce the antibodies. The T cells which are used in the activation and the proliferation of B cells, they are called as the T helper cells. The second type of the response, that is the cell-mediated response, is mainly mediated by a class of lymphocytes called as the cytotoxic T lymphocytes, or they are also known as the killer T cells. So, the basic function of this T cell is to recognize the foreign antigen. The cell-mediated response is mainly elicited by intracellular pathogens like the viruses which infect body's own cells. Because of the viral infection, these viral antigens, they are displayed on the surface of the cells, and these antigens which are displayed on the surface of the cells are now recognized by T cell receptor. Now these T cells are equivalent to the antibody producing B cells. Now let's discuss what are T cell receptors. So T cell receptor is an antigen receptor molecule which are mainly present on the surface of T cells. What is their main function? They are mainly responsible for recognition of antigen presented to the T cell by class M.Sc. class molecule. It leads potentially to the activation of T cells and elicitation of an immune response against that antigen. So this is the structure of T cell receptor. It consists of two chains, alpha and the beta chain. These chains were divided into two domains, variable regions domain and the constant regions domain. In addition to that, it also consists of transmembrane region and the cytoplasmic domain. So this T cell receptor, they are heterodimer because they consist of two transmembrane glycoprotein chains. The alpha and the beta chains having two domains, and these two domains, they are mainly linked by a disulfide bond. So this TCR, it having structural similarity to that of the Fab fragment of an antibody molecule. Now let's discuss about the structure of TCR. So this TCR, it is a disulfide link membrane anchored heterodimeric protein, and it mainly consists of highly variable alpha and beta chains. Each chain will compose of two extracellular domains, variable region and a constant region domains. Transmembrane region, they consist of positively charged groups. The variable domain of the TCR chain, they have three hypervariable or the complementarity determining regions which are also labeled as CDRs. So the variable region of the chain have an additional area of the variability, hypervariability which do not interact with the antigen and therefore they will not be considered as complementarity determining regions. While the constant domain of the T cell receptor, it consists of cysteine residues which form short connecting sequences via formation of disulfide bond, and this will link the two chains of the T cell receptors. So here you can see this is the alpha chain and this will be the beta chain. Now, the first half portion will indicate the variable region while the second one will indicate the constant region. Within the hinge region, as you can see, the disulfide bond is present because within the hinge region it mainly consists of mainly cysteine residues and therefore disulfide bond will be formed and this will help in linking of two chains. After this extracellular domain, this will be followed by a transmembrane region and in the intracellular domain, cytoplasmic tail will be there. Now, the alpha and the beta chains on the extracellular domain, they will consist of variable region and the constant region domain. Now let's discuss about the accessory molecules. So, what is the main function of this accessory molecule? They help the T cell to produce a response against a specific antigen molecule. So, what kind of the accessory molecules are there? CD3 and the zeta chains. Now, this CD3 and zeta chains, they are mainly associated with T cell receptor and their main function is to carry out intracellular signaling transaction. The second accessory molecule, that is the CD4 or the CD8 markers. CD stand for the cluster of differentiation. CD4, they are normally associated with class 2 M.Sc. molecule while CD8, they are associated with class 1 M.Sc. molecules. CD28, it will act as a co-stimulatory receptor. And the last one, that is the integrin, it is help in the adhesion. So these are the different accessory molecule. Now this is your T cell receptor. Its main function is to recognize the specific antigen. Now CD3, CD4, CD8 and CD28, they help mainly in the signal transduction process, and the last one, that is the integrin, it helps in the adhesion. So let's discuss about the co-receptors. So when the signal from the T complex is enhanced because of the binding of these M.Sc. molecules by specific co-receptors. So these helper T cells or the regulatory T cells, for them CD4 is the co-receptor and it is specific for MSC class 2 molecules. That is, antigen presented by class 2 MSC molecule will be cytotoxic T cell. The co-receptor is CD8 and this co-receptor is specific for class 1 MSC molecules. So, on the extracellular surface, these co-receptors, they help in the binding of this TCR to class 1 or class 2 MSC molecules. And because of presence of co-receptor, the affinity of this TCR to MSC will be increases and this will be helpful in cell to cell interaction between the antigen presenting cell and the T cells. While on the intracellular side, these co-receptors, they help in the recruitment of essential molecules which are mainly involved in the signaling of activated T lymphocyte, so that this will facilitate the CD3 signal transduction mechanism on the intracellular domain. So this is the structure of TCR CD3 complex. We have already discussed the structure of TCR. It consists of two chains, alpha and the beta, with two immunoglobulin like domain, V and the C domains. Now let's discuss about the structure of CD3 molecules. Now this TCR is mainly composed with or associated with multiple CD3 dimers. So one of the CD3 dimer it is consists of two chains or one is of epsilon and second one is of the gamma. It also consists of a single immunoglobulin domain on the extracellular space, a transmembrane region which span the CD3 molecule through the plasma membrane and the cytoplasmic domain. On the cytoplasmic domain, it mainly consists of ITAM, that is the immunoreceptor tyrosine base activation motifs. The second CD3 dimer, that is it mainly consists of epsilon and the delta chains. Similar to the first dimer, it also consists of a single uh, variable region or the immunoglobulin fold-like molecule on the extracellular space. Then plasma membrane will be spanned by the transmembrane region and the cytoplasmic tail will consist of ITAM, that is the immunoreceptor tyrosine base activation motif. And the third complex will consist of two zeta chains. These zeta chains are also embedded with the immunoreceptor tyrosine base activation motifs. What is the main function? They help in signal transduction. So CD3, that is the cluster of differentiation 3, it is a co-receptor and it helps in activation of the T cells. This CD3 it mainly consists of a complex of protein and they are composed of distinct chains. In mammals, the complex will consist of three CD3 chains. These chains they are associated with T-cell receptors and the zeta chains. So these CD3 chains, while their association with the TCR and the zeta chain, it is essential for the generation of signal in the T lymphocytes. So the TCR, zeta chain and CD3 molecules, they together constitute the T cell receptor complex. Let's discuss about the interaction between the CD3 and the TCR molecule. So, CD3 gamma, delta and the epsilon chains they are highly related with the cell surface proteins of the immunoglobulin superfamily. So they mainly consist of a single extracellular immunoglobulin-like domain and it is rich in the aspartate residues. While the transmembrane region of this CD3 chain, they are negatively charged and therefore they will be associated with positively charged TCR chains. The intracellular chain of all the CD3 molecules, they consist of a single conserved motif known as immunoreceptor tyrosine base activation motif or ITAM. This ITAM they are essential for the signaling capacity of this TCR. So let's discuss about the signaling and the regulation of this TCR CD3 complex. So the engagement of MSC peptide by this TCR it leads to the or required help of CD4 or the CD8 co-receptors. So these co-receptors they bind to the invariant region of the MSC molecule. So within this LCK, which is a protein tyrosine kinase, it will also act as a co-receptors. Its main function is to block the cytosolic tail of this TCR complex in a close proximity to that of the ITAM. So it mainly carry out the phosphorylation of tyrosine residues which are mainly present on immunoreceptor tyrosine based activation motifs. So this phosphorylated tyrosine residues within these ITAM sequences of the zeta chain, they will provide docking or the binding site so that protein tyrosine kinase called ZAP70 will attaches to it and it becomes activated. So here you can see once this but the first step is the engagement of MSC peptide. It will initiate the assembly of signaling complex. So these CD4 or the CD8 they will act as a accessory molecules and this is the structure of TCR and these all are the CD3 molecules. Okay. Now once this antigenic peptide recognized by the T cell receptor then the activation signal will be provided. So here you can see LCK, it play important role. So this protein kinase, it carry out the activation of the tyrosine residue by providing phosphate group to them. So once it getting phosphorylated, the tyrosine residues which are present on the ITAM sequences that are getting phosphorylated, they will provide binding site for ZAP70. Once this ZAP70 is getting activated, it helps in multicellular signal transduction pathways. So one of the pathway is the PLC mediated pathway. Second one is calcium mediated pathway. It also leads to activation of small G protein and which mainly control RAS pathway. So these pathways which was activated by this TCR, they mainly changes the expression of the genes. There may be a functional change will be observed. It controls the differentiation and the activation of various cells of the immune systems. So ZAP70, when it get phosphorylated, then it carry out the activation of large number of membrane associated adapter molecule. So we can see ZAP70 will act as an anchoring point for the recruitment of several intracellular signal transduction pathways. So one of the pathway it mainly involves the enzyme phospholipase C. Now this phospholipase C is anchored to an adapter molecule and it will get activated by the phosphorylation. So once it get activated, it will cleave the membrane phospholipid and therefore it helpful in generation of second messengers. It also have a tendency to activate the small G proteins. So this TCR CD3 complex it leads to the generation of multiple intracellular signals and many signaling pathways they are getting activated as a consequence of the step which take place within the initial phase of the signal transduction.